Hi everyone, I am here to discuss a module of course code BOC 109, course title Molecular Biology and Genetic Engineering. Here the title of the unit is the structure of DNA and RNA genetic material. Name of my module is Organization of DNA, Prokaryotic, Viruses and Eukaryotes. Now in this module, the outline you will learn is defining the genome and its types, organization of prokaryotic genome, organization of eukaryotic genome, organization of viral genome, and diagrammatic sketches of these genomes. At the end of the module, you should be able to define the genome, to describe the organization of prokaryotic, eukaryotic, and viral genome, to compare the three types of genomes, memorize the diagrammatic sketches of these genomes. Now, what is a genome? Genome is entire hereditary information of an organism which is encoded in either DNA or RNA. The name genome was first coined by Hans Winkler in 1920. There are four types of genomes. You have prokaryotic, for example, bacteria, eukaryotic, example, all animal cells, viral, example, phages, organelle genomes, example, chloroplast. Now let us see what is a prokaryotic genome organization. I told you all example is bacteria. A bacterial cell is depicted here in the slide. The genome is a circular double-stranded DNA packed in a region called nucleoid, since it doesn't have a well-defined nucleus. Length varies widely and is in the form of loops and a few million base pairs. Nucleoid also contains RNA and proteins along with 60% of its DNA. Some prokaryotes have circular loops called as plasmids, which are seen here in the figure. What are these plasmids? Smaller loops of DNA that are not essential for normal growth. These plasmids are exchanged to receive beneficial genes which are incorporated into the chromosomal DNA. Antibiotic resistance is one trait that often spreads through bacterial colonies by exchange of plasmids. So these are the plasmids here which are seen in the figure. What is supercoiling? Now the bacterial cell is quite small and the genome size varies. For example, in E. coli, it is 4.6 million base pairs. This large size of bacterial DNA still fits into the small bacterial cell due to a process called as supercoiling. DNA supercoiling refers to overwinding of a DNA strand. Supercoiled DNA is coiled more tightly than in a typical cell. Nucleoid proteins help to maintain the supercoiled structure of DNA, and supercoiling is important for DNA packing within these cells. There are two types of supercoiling structures, toroidal structure and plectonema. Now you can see in prokaryotes, plectonemic coils are more predominant because they have circular DNAs and they have small amount of genetic material. You can see the two structures here. This is a toroidal structure and this is a plectonemic structure. So this structure is more common in prokaryotes. Now going to the organization of eukaryotic DNA. Eukaryotes have much more DNA than prokaryotes. Genome is usually double-stranded linear DNA molecule. They are located inside the membrane-bound nucleus. Each species of eukaryotes has a characteristic number of chromosomes in the nuclei of its cells. Now, the letter N is used to represent a single set of chromosomes. For example, gametes like sperm and egg are designated as 1N and they are called as haploid cells. Whereas the somatic cells or the body cells are 2N because they have two sets of chromosomes and they are called as diploid cells. In human beings, the somatic cells are diploid. Human beings are diploid organisms, 2N, having 46 chromosomes in the somatic cells. Whereas the gametes, egg and sperm, are haploid, 1N, having 23 chromosomes each. We got to know what is a C-value paradox. Now, the total amount of DNA in a haploid gamete is known as the species C-value. There is no direct relationship between C-value and structural complexity of an organism. For example, amoeba has much more genome than human. So, the structure of amoeba and human beings is not comparable. Okay, so there is no direct relationship between C-value and the structural complexity. We know human beings are much more complex than amoeba. This situation is called as C-value paradox. Now, eukaryotic cells reproduce in 4 phases, G1, S, G2, M. Eukaryote chromosomes consist of a large linear DNA molecule, about 3 million base pairs. DNA must be tightly packed inside the cell's nucleus. So, this complex packing strategy to fit the DNA inside the nucleus is back again supercoiling. Now, here you can see during some stages of the cell cycle, the long strands of DNA are condensed to compact chromosomes. You can see these longer strands. They undergo all these changes and they form compact chromosomes which are having 2 sister chromatids and looks like an X, letter X. Okay. Now, structure of chromatin. Structure of chromatin in a tightly packed chromatin material is consisting of DNA and proteins. There are 2 major types of proteins associated with DNA. They are the histone proteins and the non-histone proteins. Both proteins play an important role in determining the physical structure of chromosomes. Histones are more abundant in chromatin. These histone proteins are small basic proteins with net positive charge. This facilitates their binding to the negatively charged DNA. Histones are evolutionarily conserved proteins that form an octamer of 8 histone proteins attached together. Now, these 8 histone proteins you can see in this diagram they are positively charged. They are wrapped tightly around, the DNA is wrapped tightly around the histone core of octamer histones. DNA is wrapped around histone proteins to form a structure which is called as a nucleosome. Now, this particular structure is called as a nucleosome. You can see in the next diagram, in the next picture here, the nucleosomes are wound and they are linked together by some linker structure. This is called as a DNA linker and it looks like just beads on a string. So, this structure of the DNA wrapping around the histone proteins forming nucleosomes looks like beads on a string. So, it is referred to as beads on a string structure. Chromatin fibers are usually 10 nanometers or 30 nanometers. When it is 10 nanometers, it is less compact and it shows beads on a string structure which you have just seen, nucleosomes. 30 nanometer fiber is more compact. Nucleosomes themselves become 30 nanometer in diameter. So, here 30 nanometer chromatin we have in 2 models, solenoid model, irregular zigzag nucleosome model. You can see here I have shown the chromosome here X-shaped, which is the final stage. Okay, and I am just unwinding the chromatin here and I have just enlarged the chromatin here. Okay, so here you can see the histone proteins which are wound by the DNA strand and this forms here the model is solenoid model. You can just see the solenoid model here. You can see the nucleosome model also here. So, this nucleosome model is usually seen in 10 nanometer chromatin fiber and this is seen in 30 nanometer chromatin fiber. You can see here DNA linker, you can see the histone core and you can see the wrapped DNA strand. What is a protein scaffold? Here you can see in the diagram in the figure that there are some loops which are radiating like petals of flower on the main chromosome scaffold here. Now here stretches of DNA called scaffold associated regions or SARs as we say bind to non-histone proteins to determine these loops. Loops of DNA with characteristic X shape of paired sister chromatids attached to a protein scaffold. Looped domains of DNA is held together at its base by these non-histone proteins. We already said there are histone and non-histone proteins. So, what are these non-histone proteins and their role? Non-histone proteins are less abundant. They are acidic with a net negative charge. They play a role in DNA replication, DNA repairs, transcription and recombination. These loops, as we said, radiate like loops of a flower petal, and this type of scaffolding produces 10,000 times shorter and 400 times thicker DNA than the normal one. That's how it helps in supercoiling. Now, to sum up the process of eukaryotic genome, we have this double stranded. We have just enlarged a nucleosome here. This is here the structure, you can see beads on the string. This is the solenoid model. and you have the tightly packed chromatids in the egg-shaped chromosome, metaphase chromosome. The similar diagram you can see here, a DNA, nucleosome, beads on string, solenoid model, means this beads on string is 10 nanometers, solenoid model is 30 nanometers fiber, chromatin fiber, and then you can see the interphase chromatin, and then finally in the metaphase when the chromosome is having X-shaped structure with two sister chromatids and the chromatin fiber is tightly packed with the structure of which we have discussed. Finally, we come to the organization of viral DNA, which is more simpler. The genetic material in viruses may be double-stranded DNA, single-stranded DNA, double-stranded RNA, or a single-stranded RNA. So, accordingly, they are called as DNA viruses, RNA viruses. It may be circular or linear. You can see here in the figure. Then genome includes both genes and non-coding sequences of DNA and RNA. Now in this diagrammatic sketch, you can see viral genomes, either DNA, RNA. It could be DNA linear, it could be DNA circular, okay, linear RNA, circular RNA. Accordingly, we call it as SS, DS, SS, DS, and so on. Examples of these are, you can see here parvovirus, herpes virus with linear double-stranded, circular single-stranded M13, circular double-stranded DNAs, baculovirus. Then you have RNA viruses, linear single-stranded is TMV, tobacco mosaic virus. Then you have double-stranded linear RNA genetic materials, reovirus. You have RNA circular single-stranded hepatitis, and here you have circular means any other kind of viruses. Viral genomes are expressed as translated by messenger RNAs that are either nucleic acids of positive sense or negative sense, whether they are RNA or DNA, single-stranded, double-stranded, positive or negative. First, they converge on the mRNA stage to be translated. See, the viral phage is shown here. They have got an outer protein coat here. They have got a neck and the tail fibrils. The genome is here. Whenever it has to be, when the encapsulated virus particle of this kind enters a susceptible host cell, the genome must be released from this relatively stable capsid and it must take over the host machinery. Once the genome becomes available, it can be translated directly if it is positive or negative sense single-stranded RNA, or else it has to go to the formation of mRNA. So that the process will commence. Now here in this diagram, in this figure, we can see, in this picture we can see the phage is infecting a cell and then takes over the host machinery, produces its own, and then there is lysis of the cell and release of the viral progeny. These are some of the references which you can refer to. Hope you have benefited from this module. Thank you.